Hi everyone and welcome to the WebUnafi channel. I'm Dr. Najwa bin Faraj from the Department of Innovation in Biological Agro-Food and Forest Systems at the University of Tusia in Italy. This video entitled Phytophthora citrus diseases is a part of our activity carried out within the project WebUnafi from waste to bio-based nanoparticles for the management of Phytophthora diseases of citrus funded by the European Commission. Let's begin first with an introduction of the genus Phytophthora. Phytophthora are microorganisms that belong to the Omicota division. As shown here in this tree, Omicota, including the genus Phytophthora, were classified in the past as true fungi, and this classification was based on the similarities found between Omicota and true fungi. One of the similarities between Omicota and true fungi is that both of them are eukaryotic microorganisms, which means their cell contain true nucleus enclosed within a membrane. And this shared cellular complicity sets them apart from prokaryote organisms, like for example, like the bacteria. And it was one of the reasons that they were initially grouped together in the first classification. Under microscope, both exhibit a filamentous growth during their vegetative stage called hypha. Also, as you can see here, another similarity between Phytophthora species and the true fungi is that both of them have the same colony morphology. As you can see here, the colony of Phytophthora and those of true fungi often appear visually similar when grown on the media culture. Furthermore, those microorganisms obtain their nutriment by absorbing organic compounds from the surrounding environment. However, and recently and based on phylogenetic analysis, scientists have discovered that Phytophthora is actually more closely related to the algae than the fungi. And this surprising relationship led them classify the Phytophthora separately than the true fungi. Another important biochemical difference between Phytophthora and the true fungi lies in the composition of their cell walls, because the true fungi cell walls are primarily made by the chitin, which is strong nitrogen contains polysaccharide also found in the insects, and this chitin gives the fungal cell wall their rigidity and resistance to the environment stress. However, Phytophthora, as the other Omicota species, have cells that are composed mainly by the cellulose, by the beta-glucanase, especially the glucose polymerase. And this is more similar to the cell wall structure of plant and algae than to the fungi. According Phytophthora is now placed in diverse group of eukaryotic microorganisms that include the brown algae and water molds. And within this kingdom, Phytophthora now are belong to the Omicota species or Omicota phylum, which are fungi-like but not true fungi. About the life cycle of Phytophthora species, Phytophthora produce both the asexual and sexual reproduction stages, allowing it to rapidly spread and persist in challenging environments. In citrus, in citrus crop, Phytophthora constitute a dangerous pathogen. Before talking about Phytophthora citrus disease, I would like further to explain a fundamental principle in phytopathology, which is the disease triangle. So for the expression of any plant diseases, we need the interaction between three critical factors: the virulence of the pathogen, the susceptibility of the host, and the existence of environment, favorable environment condition. So when those three elements occur together, a disease can develop. In the case of the citrus, the most common Phytophthora species that are responsible for several infections are Phytophthora citrophthora and Phytophthora nicotiana. Those Phytophthora species can infect almost every part of the citrus tree, including the roots, the trunk, the limbs, and fruits, leading to significant crop damage and decline. One of the most dangerous Phytophthora citrus disease is the citrus root rot. The infection by Phytophthora begin in the roots, and the roots after the infection became soft and dark due to the enzymatic degradation of the cell wall of the plant due to Phytophthora. And after the infection, the, the tree become how to say become will have a problem because it will not be able to absorb water or nutriment, leading to symptoms such as leaf yellowing, wilting, and over decline in vigor. And this kind of tree that are suffering from these diseases will be more susceptible to secondary infection and environment stress and may eventually die. And this in this table you can see the significant economic threat of citrus crop due to these diseases. Another important disease is the gomosis, or also called the foot rot, uh, that is also a dangerous citrus disease. And this disease typically begins near the crown of the tree or just below the soil line and can extend upper along the branch and or the trunk. And one of the most characteristic symptoms is the presence of crank in the branch from which dark amber colored gum appear, and from here comes the name gomosis. The last Phytophthora diseases that we will talk about is the citrus brown root that pose a major threat both in pre and post-harvest fruit quality. And the infection typically became when the zoospore from, for example, the soil became in contact with the fruits, especially during the period of high humidity and excess soil moisture. Thus, the objective of the project was BioNafi, which is from waste to bio-based nanoparticles for the management of phytophthora diseases of citrus, is the exploitation of agriculture waste as a resource for the synthesis of new nanoformulation of biofungicides based on nanoparticles charged with essential oil and fungal metabolite against citrus brown root and gomosis caused by Phytophthora species. To reach this other objective, the project focused on the survey on the current management strategies for controlling Phytophthora associated with citrus disease and waste, the evaluation of potential of agriculture waste in the synthesis of essential oil as source for a biological control agent, in vitro evaluation of antifungal activity of essential oil and fungal metabolites against the causative agent of fruit brown root and gomosis of citrus, the preparation and the characterization of nanoparticle dispersion and in vitro tests, in vitro effect of alginate nanoparticle against fruits brown root and gomosis, and then the life cycle assessment of the new formulation of of the bio formulation. For more information, please don't hesitate to contact us by mail or using platform of the project like YouTube, Facebook, Twitter, Instagram, and LinkedIn platform. Thank you for your attention.